Good morning, students. Am I audible?
Good morning, ma'am. Yes,
Good
ma'am.
morning, ma'am.
Okay, fine. So today we have finished the diagnosis part and we are going to move on to the management part now, right? So in management also it's divided into further the management of a plant viruses, then management of a human viruses, and then management of the animal viruses. So most probably 3 or 4 lecture will go on the management topic, right? So management I have discussed. I mean, from this beginning only when I started with the diagnosis, I briefed about the management also. That management of viruses when they are already in the field, when we talk about plant viruses, is not possible. So we need to look for some other methods by which we can prevent the pathogen to, excluding the pathogen from the field. So what are those methods that can be used? So in management, we'll discuss the general disease management or integrated pest management or a general principle of a disease management. So which covers all the pathogen, and then we'll move to the methods which are based in case of a plant viruses, right? So we should start. So plant disease management.
Ma'am, your voice is breaking.
Yes, ma'am.
Yes, ma'am, your voice is breaking.
मेरे फोन पे भी hello, am I audible now? Yes, the Wi-Fi is actually very slow today, so uh, maybe for some time, if there's some problem, if you're not able to hear me, at least to message me, or just wait for me if I'm out of the meeting, because due to the network, I might leave the meeting also in between, right? So when we talk about virus disease management, we always talk about the integrated approaches, so that all we can use. Why integrated approaches? So that all possible ways for the management of any disease can be used. So what is the use of integrated approach basically? Um, when we are using an integrated approach, we are using environmental friendly methods, or those methods where we don't use chemical first. So we try whatever other methods are available other than the chemical control, because chemicals are the environment pollutants also, and they definitely have some kind of a just a moment. So chemicals, they have a negative impact on the environment also, and on human and a plant and animal health also. So before we opt for a chemical control, we try other methods such as a biological control, or some cultural practices, or such preventive methods such as we use a virus-free or a pathogen-free starting material. So other methods are being used before we go for the chemical control. So chemical control is always the last option when we don't have any other control for the management of a particular insect, pest, or a disease. So in an integrated approach, there are 4 principles. First is the exclusion, then the plant resistance, because in naturally also the plants, they have sometimes resistance genes which are for some pathogens, so we can exploit those resistant genes, or we can exploit those resistant varieties and can be use them. Then cultural controls include the practices such as leaving the land fallow for a certain time, and then not repeating the same kind of a crops every time, and maybe the soil solarization or other methods that can be used, and at the end the chemicals. So chemicals are basically in the bottom, that when no other method is being successful, then we can go for the chemical control. So exclusion can be done by regulation of a planting material at the ports, a city or a national or international boundary. So that means the disease materials should not be taken out of the country, or within the country also. There are actually certain regulations where the planting material or seeds or other things cannot be taken from one part to the other. They need to be checked, or they need to be certified. So pathogen free seeds or a plant, so seed certification is one method. Then meristem tip culture, this we have, I guess, discussed many times that meristem tip culture is one method by which the pathogen-free plants are being produced. So again, if plants are being generated by meristem tip culture method, that means they are definitely 80 to 90% chances they're free from the pathogen. Then cuttings, those plants which we grow through cuttings, so cuttings from clean mother plant under sterile conditions. So if the stock plants or the mother plants are a virus infected or pathogen infected, there are chances, definitely there will be chances that the plants that we use, those plants will use for cutting and growing further the plants for the propagation or a multiplication, they will also be a carry the pathogen with them. So cuttings from a clean, we should make sure that the stock plants or a mother plant should be free from any kind of a pathogen. Then resistance, plant resistance, mm, genetic immunity is again the role in the plant kingdom. So if immunity does not exist, plant breeders, they develop cultivars with resistance to specific pathogen, right? So this is basically done, the some of the wild, whatever cultivated plants we have, they have some kind of a wild relatives which are being grown naturally out there, maybe in untouched flora, right? So we can exploit those plants, we can study those plants, and we can see whether they have some kind of a plant resistance or pathogen resistance gene in them. So we can exploit those variety, we can use those variety for growing. Then adaptation, plant adaptation to a site. So it is also very important that when we are growing plants, they should be well adapted to one particular location. So that also provides them some kind of, if they are well adapted to one location and the pathogen free environment is there, or maybe sometimes the vectors of those pathogen are not available in that environment, so this method can also be used. Then plant resistant, if we talk about the genetic resistant, it is again a constitutive and an inducible two type. So continuous defenses, for example, some plants, they build in such a way that they have some kind of a continuous defenses present. For example, they have a cell walls. So in order to penetrate inside the plant, viruses, they need a certain kind of a vectors, or they need a certain kind of a injuries, otherwise the cell wall cannot be broken very easily. Then epidermal cuticles are also there, there's a bark on the plant, the leaf hairs, right? So there are certain kind of a physical and a chemical barriers which are present in the plant, so they also prevent the pathogen attack on the plant. Then inducible are basically defenses which can be induced by chemicals or certain kind of a proteins that we can use for them. So these are inducible basically defenses which are produced in response to the invading pathogens, right? So include toxic chemicals or a pathogen degrading enzymes, or certain sometimes the deliberate plant cell suicide also due to the infection of a particular. What happens, when certain pathogen they infect plant, right? So certain kind, or sometimes around that area, around that cell, the plants, they make those cells die, right? They become necrotic. Necrosis is being generated by the plant's defenses also. So that area become dead, so that pathogen is not able to move further. Then we have a systemic acquired resistance. So this kind of a resistance is activated when pathogen infect tissue, and this is a long lasting systemic immunity, even in tissues not infected. It further moves. Systemic means throughout the plant. It is relatively broad spectrum and associated with increase in phytohormone salicylic acid. Then chemically induced systemic acquired resistance. So some systemic acquired analogs can induce systemic acquired resistance like responses also, which provide resistance in plant tissue beyond the application site. That is systemic resistance is finally. These are referred as plant activators such as benzothioactigard. Actigard is basically one which comes under the sales name in the market, which is used for the plant protection. Then adaptation, so right plant for the right site. So this we all know actually, when we tropical plants cannot be grown in the temperate area and vice versa. So right climate is plants are adapted to a different kind of a climatic conditions, right? So we know in a temperate plants they require a certain kind of a cold treatment, so those plants cannot get that treatment in the tropical areas or warmer areas, so they cannot be grown there. So temperate was tropical, cold induction, dry was rainy, those plants which need a lot of rain, whereas those plants or plants who have adapted them to the dry like or a desert like conditions, right? Then a soil type also, that what kind of a soil they can make, day length and everything. So genetic resistance can be overcome if site is not right for the plant species. So if we are growing a particular plant out of one place and we are growing it in some other place where the environment and the weather factors or weather variables are not actually the which is in which in those which are required for the plant to grow, so the genetic resistance can be overcome in those areas. Then cultural control. So this is again this is one kind of a control. Basically, we don't require much. This is just the the how efficient farmers are in understanding this. So basically, someone need to tell the farmers that okay, these are the cultural practices that you can follow in your field. So could be the crop rotation. Crop rotation means not growing the same kind of a crop in the field again and again. So that establishes a particular pathogen. It is a host specific in the field, and then it will live there for longer time. So crop rotation is one another factor which we can utilize to manage the pathogen. Then alternate host eradication. So what does this mean? Pathogens, the crop they affect, they are the main host for the pathogens, but sometimes they live on the alternate host also, and whenever they find the suitable crop which is the main host, they move to those kind of a host. So we can remove the alternate host or a weed reservoirs from the vicinity of the crop so that they don't get a chance to go and stay there on the alternate host. Then sanitation for tools, equipment. This is very important because the pathogens they get transmitted by this method also. Then improve plant environment, water management, and the air circulation. So proper spacing between the plants should be there. Nutrient management. So nutrient deficient soils they invite more pathogens. So proper nutritions or nutrients which are required for the plant growth should be provided. Soil treatment such as solarization. So soil solarization is basically a practice where the fields they are covered with the plastic sheets, maybe for a 1 week or maybe for a longer duration. So those plastic sheets they kind of build the greenhouse effect. So they'll the those sun rays they'll be trapped inside those sheets, and that will raise the temperature of the soils much higher. So those higher temperatures will kill the any remains of the vectors or any pathogens, spores or whatever they're present in the field. So that will make the soil sterile. This is one common method again which is being used. Chemical controls are many. There are seed treatment, there are soil treatment, root trenches. So in viruses, the chemical control is usually only for the vector management. So sometimes we need to use some insecticides or pesticides to manage the vectors which are transmitting the disease. But otherwise, as for viral diseases, we don't have any chemical control available with us, right? So I'll just escape this chemical control. Just then microbiological control. Again, microbiological control is present for fungi, certain fungi and bacteria. Some species they are able to manage or kill other pathogens, especially the insect pests or maybe other organisms. So that is called a microbiological control. But for viruses, microbiological control is not there. Okay, this is one Bacillus subtilis strain which is present in the market, which is used as a biopesticide. Then plant disease management, again, this is a similar approach. We are talking about the plant viruses, exclusion, resistance, culture, chemical, and microbiologicals. So GMOs, genetically modified organism, it definitely comes in an integrated disease management program. But with GMOs, there are a lot of issues, societal and ethical issues, which are associated with the GMOs, but still genetically modified pathogens or developing plant resistant against the viruses can be done by using transgenics, which is a genetically modified organism approach, right? So this now will start with the plant virus disease management. Earlier method that I discussed, they are common for different plant pathogens. Some are applicable in case of plant viruses, where others are applicable mainly for bacterial or other pathogens. So prerequisite for any plant management are the first, as I told you, like correct pathogen ID. So that means the diagnosis. That was the previous lecture that we did. So correct diagnosis for the management of any plant pathogen is very important. Then understanding pathogen biology or disease epidemiology, which discuss about the what kind of a pathogen is this, what are different environmental factors which are affecting the pathogen, right, or a vector or other things. And then development and evolution of a management strategy. So after that, we develop a strategy for the management, that how we can, using different approaches, we can manage a particular pathogen. So what are viruses? Let's just go through this, because we all know viruses. What are viruses? So viral disease management falls under conventional approaches and biotechnological approaches, two broad categories. So when we talk about conventional methods, indexing and certification program is the same as I told you earlier, that certification of a planting material before they are being used in the field is very important method, where virus-free plants can be grown in the beginning, right? Heat therapy is done against potato virus. Why? So sometimes heat treatment of the seeds or the tubers of the potato can be done to inhibit the virus infection. Meristem tip culture is again a plant tissue culture technology by which we can develop a virus-free plants. Then there are certain oil sprays. Oils can be sprayed on the plant, and these oil spray can prevent the vector feeding on that plant. So in this way, if vector is not feeding, because the virus transmission is basically taking place mainly through the vector. So if the transmission, that vector is not able to transmit to the another plant the virus, so that way also we can manage. Cross protection, I'll discuss this strategy in little detail, that how we go for cross protection. Then barrier crops, sometimes in between the smaller crop, bigger crops are being grown somewhere, so that the vectors, they cannot move from one plant to the other plant. So such barrier crops can also help in preventing the virus transmission. Then reflective mulches in case of a squashed mosaic virus. Sometimes some mulches means you cover that particular area with certain mulches that reflect the light. So insects or a pest which are feeding, or which are the vector of those particular virus, they are not able to feed on the plant for a longer time due to the reflection which is caused by that. So that also help in preventing the virus transmission. Then the natural resistance. So some plants are naturally resistant to certain kind of a pathogens due to certain genes present in them. So that can be done. So indexing and a certification means removing crop residues or remnants of a virus infected plant, which can act as a primary source of a virus infection. So prevent direct contact of a healthy plant with the already virus infected plant, or a contaminated hands, or whatever tools we are using, right? Then we remove the perennial or annual weeds which act as the sources of virus in disease, legumes and cucurbits, and the use of virus-free certified propagation material. So stock that we are using for a propagation should be virus-free. So whatever plants we are developing through these strategies, they should be virus-free. Virus-free seeds should also be used. So that means seeds which are seeds or a planting, other form of planting material which is used for growing a particular plant or a crop, should be tested beforehand for the presence of any kind of a virus. So there are certain referral labs which are present throughout the country to do these kind of a testing. So on a minimal prices, they do this testing, and then that planting material is being certified. So if they found any kind of a virus on that, they'll name that this virus is being found and others are negative, or if there's no virus being found, so they'll say it is certified that it is free from all kind of the major viruses which are expected to be infect that particular crop. Then cultural method, susceptible seed beds can be covered with tall barrier crops to prevent the spread of the virus. Then growing susceptible plants in isolation away from the other plants, alternation of a date of sowing or a changing planting date. So little, if we, in order to prevent or in order to escape the vectors, we can do the little early sowing or little late sowing also. So that will also help in prevention of the virus. Growing plants in a sterilized soil. Okay, the sterilized soil, this can be done in a smaller way, for example, in a glass houses or greenhouse cultivation. But when we are going for a larger wheat fields or a rice field, this doesn't seems to be feasible, right? And then leaving fields fallow and a crop rotation. So fallow means if this season I've grown a tomato in a particular field, so next season I leave it as such. I'll not grow anything in that field, or if required, I'll grow another crop, not the tomato. So that will again help in somewhere the prevention of the pathogen. Then heat therapy. So this is or thermotherapy is a very important method which is being done in a plant virus management since lot of since past many years. So thermotherapy combined with the chemotherapy or other method, they are doing it, right? So hot water treatment of a dormant provocative organs such as tubers or a bud roots, which we use for growing plants, they are dipped in a hot water, 35 to 54 degrees. It depends on the kind of a basically we know the thermal inactivation point of every virus varies. So according to that, it can be changed. So hot treatment for a different intervals, actively growing plants in a growth chambers, 35 to 40 degree for several weeks helps in eliminating the virus from the plant. Potato leaf roll virus can be freed from potato tubers if they are stored during summers and in temperature up to 36 degrees Celsius. So this has been tested. Then meristem tip culture. So again, this is a plant tissue culture method which is used to produce virus-free stock. So apical meristem is a region in the plant which is free from the pathogen or a virus in general, right? So 0.1 mm to 0.5 mm tip, and then if it is grown in vitro tissue culture medium to raise the virus-free plant. So ingredient tissue culture, we need a tissue culture media and a proper growth hormone and a light condition so plants can be done. So this technique works well for chrysanthemum, carnations and potatoes also. Then chemotherapy. So chemical control of viruses has not been very successful. This is from some experiments. So when they did some treatment of a tobacco plant with a virusole, so that delayed or prevented systemic infection with tomato spotted wilt virus. And chemical carbendazim, which is present in the fungicide Bevestin, it also reduces virus-induced symptoms, but then it has no impact on the viruses. So such symptomless host can be, symptomless hosts of a viruses are more dangerous because when we see that there is no symptom present on the particular plant, we assume that it is virus free. But then since they don't asymptomatic hosts which are present there, but they are going to transmit the virus, right? So they act as a reservoir of a viruses for other plants. Chemical treatment by itself has not yet found practical use. However, chemical treatment in combination with heat treatment or a meristem culture, they have some advantages in few instances or few research which has been done. So plant protection chemicals, natural defense mechanism against viral infection by certain plant extract has been reported. So they show usually two type of inhibitory response. So mostly they reduce the virus activity when co-inoculated with virus into susceptible plants, for example, Dianthus caerophilus or Phytolecca americana. However, extract from a few non-host plants such as Beauveria diffusa and Mirabilis chelpa inhibit the virus infection and development of symptom by stimulating the production of virus inhibitory agents in a susceptible host. So there are some plants, we can say they are being recommended that their plant extract show some kind of inhibitory responses against the viruses. So similar virus inhibitory agent may also be induced following inoculation or spraying of susceptible plant with either virus or the chemical. So these inhibitory agents may be low molecular weight proteins or a glycoprotein and show some kind of a resemblance to the interferons. So foliar spray of oil or a skimmed milk have also been used to prevent the spread of virus disease by the aphids. So aphids, they fail to infect such plants. So some biocontrol or some plant extracts or botanicals have been used for the, in various experiments, people have tried to check their efficacy. Then elimination and control of insect vectors. So this is very important because plant viruses are transmitted by the vectors. So if we can manage the vectors, we can manage the plant viruses also. So we need to focus where we cannot do any chemical control is not there for viruses. Chemical control is sure there for the viruses. So we can use those kind of a chemical control in a recommended doses to eliminate the vectors. So avoidance of a vector in virus diseases which are transmitted by the insect. So incidence can be minimized by avoiding the contact of the vectors with the host, by growing crop in isolation where vectors are present, absent or low in number, tall cover crop to protect undersown crop from insect-borne viruses. For example, cucurbits are sometimes grown intermixed with the maize. Barrier crops can be grown around a crop or in alternate rows between crops. So they have been found useful in controlling non-persistently transmitted aphid-borne viruses. Then use of a reflective mulches, as I told you earlier, that you can use these mulches that can, avoid that due to these reflective mulches, the vectors, they are not able to feed on the plant. Then direct control of the vector can be done by chemical control using insecticides. So it can be again natural or a synthetic. Natural are some insects can be controlled by insecticides are derived from the tobacco. The nicotine in tobacco affects them. Then chrysanthemum, there's a pyrethrum in chrysanthemum which affect the vectors, and legume there is rotenone and the neem also. So neem, we all know that when we keep neem leaves with our food products, they repel the insects or the pests. So that way it can be managed for this. Synthetic pesticides are again the malathion, rocher, or damicron. They can be sprayed or applied as a soil drench or as a granules or a mix in a soil. So sometimes when we talk about the natural insecticides, so the production of these kind of insecticide, they require a lot of work and a lot of these are not very economic. So people usually opt for the chemical control, though the chemical control is somewhere directly affecting the environment and a human and a plant and animal health also. So maybe we need to work more on the development of a natural insecticides and make them at a price which a farmers can actually go for, and they can opt for it. Because when given a choice, whether you'll opt for a chemical control or a natural, they want to opt for natural or some natural products, but they are so costly that they cannot afford, whereas the synthetic pesticides, they come at a very cheaper rates. So definitely they opt for those chemical pesticides because they don't have to spend much on that, right? Okay, so I'll stop here because I have some meeting today to join. So if there's anything which is not clear till now, you can ask your questions. Oh, yes, Sukta.
Ma'am, what are fumigants?
Fumigants are okay. See, fumigants are certain chemicals which release fumes when they are released in the soil or a plant. So they also, through fumigants, are basically gaseous chemicals which are used for control of a pest in the agriculture fields, right? जैसे आप फ्यूमिगेशन करते हो ना, मॉस्किटोज के लिए फ्यूमिगेशन होती है ना, through some fumes, it is the similar kind. Okay.
Ma'am, what is virus?
What is Virozole? Is an antiviral basically, which is like we have some antiviral drugs which are present to control the human ribavirin, and the Virozole is a similar kind of antiviral drug which is present, and they say its spray can used in prevention of some viral pathogens. But then again, the Virozole is a costly drug, so we cannot afford to use in a spraying, right?
Yes, ma'am.
Okay.
How is
Yes.
its efficiency?
See, Virozole's efficiency is they've tested it for some plant pathogens, not for any. That too in the research programs only, because the kind of the doses that we use to release in or use in a particular field, that will be more. But when we talk about in a research experiment, it is fine. Its efficiency is fine. They can control some this virus to some level, but not that much. So that's why they have dropped the idea of using antivirals, especially in a plant virus management, because first they cannot afford, second they are not that efficient that even if they are using them, still the virus infection is being reported, right?
Yes, ma'am.
Okay, students. So please, for your attendance, quickly switch on your camera so that I can take your attendance and I can go for my meeting. Yes, Adesh, Athoiba,
Present, ma'am.
Present, ma'am.
Ayushi,
Present, ma'am.
Sam,
Present, ma'am.
Devanshi,
Present, ma'am.
Ishita,
ma'am. Present, ma'am.
Janvi, Kshitij,
Present, ma'am.
Present, ma'am.
Anaswani,
Present, ma'am.
Namrata,
Present, ma'am.
Nandini, Nehal,
Present, ma'am.
Upal,
Present, ma'am.
Sahiba,
Present, ma'am.
Shakti, Shivani,
Present, ma'am.
Tanvi,
Present, ma'am.
Tridiv,
Present, ma'am.
Vivek, Gupta,
Present, ma'am.
युवराज,
Present, ma'am.
Present, ma'am.
युवराज के बाद कोई नहीं है ना, युवराज is the last one, right? Okay, students, so I will see you in tomorrow's class, right? Thank you so much.
Thank you, ma'am.